Welcome back to IGCSE Science Academy. Our mission is simple, providing clear, concise, and comprehensive lessons designed specifically for the IGCSE curriculum and for the competitive exams. Let's get started. Now diving straight into our core material for today, theories of evolution. This is a critical module for anyone preparing for CSS, PMS, and FPSC exams under the biology and journal size. Evolution isn't just one single idea, it's a framework built over centuries. In this lesson, we are going to break down every major milestone, including Lamarckism, Darwinism, Neo-Darwinism, synthetic theory, mutation theory, natural selection, genetic drift, speciation, evidence of evolution, Hardy-Weinberg law. Finally, we will wrap up with the specific FPSC key points to ensure you are ready for the exact types of questions that appear on test day. Let's start with the foundations. Before we get into the biology topic, theories of evolution, let's look at the official Federal Public Service Commission FPSC notice for the senior elementary teacher position. If you look at the test specification section, you will see the exam is broken down into three parts. Part 1 English, Part 2 subject test, Part 3 is the professional test. English carries 20 marks, subject test 40 marks, and professional test 40 marks. We are focusing we are focusing on the biology, zoology, and botany, let's discuss the topics which are included in the syllabus. Part 1 for the English grammar usage and sentence structuring. Part 2 the topics which are included for the subject test. First is theories of evolution, nucleic acid, cell division, nutrition, ecosystem, division of plants, bryophytes and Petridophytes, gymnosperm and angiosperm, ecology, cytology, genetics, ecosystem, classification of fish, reptiles, mammals, mollusks and Echinodermata, membrane system, mammalian gametogenesis and fertilization, and last, Mendelian laws of inheritance. Part 3, which is professional test. Topics are education system in Pakistan, educational psychology, classroom management and discipline, instructional planning and teaching strategies, students, evolution and examination system, educational guidance and counseling. We are focusing on part 2 under the syllabus for biology, zoology and botany. The very first topic listed is theories of evolution. This confirms that the material we are covering today isn't just extra reading, it's a core requirement for those 40 subject marks. By mastering this slide deck, you are checking off the first major box of your FPSC preparation. Let's make sure you get those marks. Table of contents. The topics which we cover today from the theories of evolution are Introduction to Evolution, Basic Concepts, Definitions and History, Lamarckism, Theory of Inheritance of Acquired Characters, Darwinism, Theory of Natural Selection, Neo-Darwinism, Modern Evolutionary Synthesis, Mutation Theory, De Vries, Role of Mutations in Evolution, Genetic Drift and Genetic Flow, Allele Frequency Changes, Evidence of Evolution, Fossils, Anatomy, Embryology and Biochemistry, and then Speciation and Hardy-Weinberg, Species Formation and Equilibrium. These are the topics which we are going to cover today. Now that we know what the syllabus requires, let's get into the actual science. What is evolution? Simply put, it is the gradual changes in heritable characteristics of a population over successive generations. It's not just about one individual changing, it's about how a whole group evolves over time to form a new species. So the definition, as we already discussed, that evolution is the process of gradual change in heritable characteristics of biological populations over successive generations, leading to the formation of new species. To master this topic, we need to be able to distinguish between these six key terms. First is organic evolution. Change in genes frequency in a population over time. This refers specifically to the change in gene frequency within a population over time. It's the biological engine of everything we are about to discuss. Second is microevolution. Small-scale changes within a species, for example, antibiotic resistance. Think of these in the term of scale. Micro is small scale, like a bacteria becoming resistant to antibiotics. Then macroevolution, large-scale changes leading to new species or higher taxa. So macro is the big picture, large-scale changes that lead to entirely new species or higher taxonomic groups. Phylogeny, evolutionary history relationship of a species or group. This is the family tree of biology. It represents the evolutionary history and relationships of a species or group. Fitness, ability to survive and reproduce in a given environment. In evolution, fitness isn't about being the strongest, it's about reproductive success. It's the ability of an organism to survive and, more importantly, pass its genes to the next generation in its environment. And the last, adaptation, heritable trait that increases an organism's fitness. Finally, we have adaptations, the heritable trait that actually increases an organism's fitness. Memorize these definitions as they often appear in the MCQs portion of the FPSC biology test. Moving on to our first major theory, we have Lamarckism, also known as the theory of inheritance of acquired characters, proposed by the French naturalist Jean-Baptiste Lamarck in 1809. This is a frequent topic in FPSC exams, so pay close attention to his four key postulates. Lamarck's work, published in his famous book Philosophie zoologique 1809, was one of the first organized attempts to explain how life changes over time. He based his theory on four main pillars: use and disuse. Organs used frequently become stronger, unused organs degenerate. Example, giraffe's neck elongated by stretching. Lamarck believed that if an organism uses an organ frequently, it becomes stronger and larger, while unused organs eventually wither away. The classic example of this theory was giraffe necks, which he argued grew longer as they stretched for high leaves. Second is inheritance of acquired trait. Changes acquired during an organism's lifetime are passed to offspring. This is the core of his theory, the idea that changes an organism acquired during its own lifetime can be passed directly to its offspring. Third is effect of environment. Environment directly causes changes in organism which drive evolution. He pointed that the environment acts as a direct driver for these physical changes. Then fourth is internal vital force. Organisms have an innate drive towards complexity and perfection. Finally, he believed organisms have an innate internal drive to become more complex and reach perfection. So criticism, Lamarckism was disproved. Auguste Weismann's germplasm theory showed somatic changes cannot affect germline DNA. Cutting trait of mice for 22 generations produced no short tail offspring. So it is important to know that this theory was eventually disproved by Auguste Weismann, famously debunked Lamarckism with his germplasm theory. He cut off the tails of mice for 22 generations, yet every single offspring was still born with a tail. This proves that somatic changes, physical changes to the body, do not affect the germline DNA passed to children. Moving from Lamarck, we arrive at the most revolutionary name in the biological history, Charles Darwin. His theory of natural selection, published in 1859, changed our understanding of life forever. For our FPSC exam, we need to master these 6 logical steps that Darwin used to explain how evolution actually works. In 1859, Charles Darwin published his landmark book On the Origin of Species. While Wilfred Russell Wallace was a co-discoverer of these ideas, Darwin is the name most associated with the theory of natural selection. Let's break down his 6-step logic. First is overproduction. All species produce more offspring than can possibly survive. Resources are limited, causing competition. Darwin noticed that all species produce for more offspring than the environment can support. If every oak tree seed grew into a tree, the world would be covered in nothing but oaks. Second is variation. Individuals in a population differ from each other in heritable traits. No two individuals are exactly alike. He observed that no two individuals are exactly same. There are always small heritable differences within a population. Number 3, struggle for existence. Competition for food, shelter, mates, predation, and diseases create constant pressure on survival. Because there are too many individuals and limited resources, food, shelter, mates, life become a competition, predation, and diseases add to this constant pressure. 4 is natural selection. Organisms best suited to environment survive and reproduce. Survival of the fittest, coined by Spencer. This is the filter. Organisms with traits best suited to their environment survive and critically reproduce. This is often called survival of the fittest, a term actually coined by Herbert Spencer. 5 is heredity. Favorable traits are inherited by offspring, gradually increasing in frequency in the population. Those favorable traits are passed down to the next generation over time. The frequency of these good traits increases in the population. Speciation. Over many generations, accumulation of changes led to new species. Common descents with modification. So over many generations, these accumulated changes led to the formation of entirely new species, what Darwin called descent with modification. Remember, Darwin's key evidences came from his observations of Galapagos finches and the fossils record. If the FPSC asks for the mechanism of Darwinism, the answer is always natural selection. Now let's discuss about natural selection and types and examples. Building on Darwin's core idea, we need to understand that nature does not always select traits in the same way. This slide covers the 3 modes of natural selection, a favorite topic for FPSC examiners because of its real-world application. So natural selection isn't just a general concept, it follows specific patterns depending on environmental pressure. To master this for our exam, we must be able to identify these 3 types. Directional selection. One extreme phenotype is favored, population shifts in one direction. Example, antibiotic resistance in bacteria, industrial melanism in prepared mouth, Biston betullaria. So this occurs when environmental changes favor one extreme phenotype over the others. The population's average shifted in one clear direction. Think of antibiotic resistance in bacteria or the classic case of industrial melanism in the peppered mouth, Biston betullaria. When darker mouths become favored as tree grew shoot cover. Stabilizing selection. Intermediate phenotype is favored, extreme are eliminated. Example, human birth weight, very low and very high birth weights have higher mortality. Here nature favors the intermediate phenotype and the extremes are eliminated. This reduces variation and maintenance that status quo. Examples, human birth weight is the perfect example. Babies with very low or very high birth weights have higher mortality rates, so the middle weight is favored by selection. Disruptive selection. Both extreme phenotypes are favored over intermediates, leading to polymorphism. Examples, Darwin's medium ground finch with two distinct beak sizes. This is the opposite of stabilizing selection. Both extreme phenotypes are favored over the immediate ones. This can lead to polymorphism, where two or more distinct forms exist in the same population. So Darwin's medium ground finch, where individuals with either very small or very large beaks survive better than those with medium-sized beak. Before we moved on, remember that fitness equals reproductive success. Adaptation is your heritable advantage, and selective pressure is the environmental force like a predator or climate driving these changes. We now move into the 20th century with neo-Darwinism, also known as the modern evolutionary synthesis. Develops between the 1930s and 1950s. This theory is the great integration. It combines Darwin's natural selection with Mendelian genetics, population genetics, and paleontology. Think of it as Darwin 2.0. The key architects you should remember for the FPSC exams are R. A. Fisher, J. B. S. Haldane, Sewall Wright, Ernst Mayr, G. G. Simpson. To understand how evolution works at the genetic level, we look at these 6 drivers. First is mutation. Random changes in DNA provide the raw material for evolution. Point mutations, chromosomal mutation. These are the random changes in DNA, point mutation, or chromosomal shifts that provide the raw material or new variation for evolution. Gene recombination. Crossing over and independent assortment during meiosis generate new gene combinations. This happens during meiosis through crossing over and independent assortment. It's why we don't look exactly like our siblings. It's shuffle the genetic deck. So then natural selection acts on phenotypic variation produced by mutation and recombination, determines which allele increases in frequency. It still plays its role acting on the phenotypic variation produced by those mutation and recombinations to determine which allele gene version increases in frequency. Genetic drift. Random changes in allele frequency in small population founder effect the bottleneck effect. These are the random changes in allele frequency that happens by chance, especially in small population. Watch out for terms like the founder effect and the bottleneck effect in your test. Gene flow. Transfer of alleles between population via migration reduces genetic differences between population. This is the transfer of alleles between population via migration. It helps reduce genetic differences between groups. Reproductive isolation. Barriers preventing interbreeding essential for speciation, geographic, behavioral, and temporal mechanical. This is the final step for speciation. Barriers, whether they are geographic, a mountain range, behavioral, different mating zones, or temporal, different mating seasons, prevent interbreeding by allow new species to form. By combining these factors, neo-Darwinism explains evolution not just through physical traits but through the changing frequencies of genes in a population. In this slide, we move to Hugo de Vries and his mutation theory from 1901. which challenged the idea that evolution only happens through tiny gradual steps. While Darwin emphasized gradual change, the Dutch botanist Hugo de Vries proposed that evolution is driven by sudden large heritable changes which he called mutation. De Vries' observation studied Onithera lamarckiana evening primrose. Observed sudden large heritable changes, called these changes mutation. Proposed mutation, not gradual change cause evolution, and the book is The Mutation Theory, 1901 to 1903. He based his entire theory on his study of the evening primrose Onionthera lamarckiana. He noticed sudden variation in these plants that were passed to the next generation, leading him to believe that new species arise in a single large step, a process sometimes called mutation. He published these findings in his book The Mutation Theory between 1901 to 1903. Types of mutation to remember. First is gene mutation, change in DNA base sequence, point mutation. A change in the actual DNA base sequence, often called a point mutation. Chromosomal mutation. Deletion, duplication, inversion, translocation. Larger changes like deletion, duplication, inversion, or translocation of chromosome segment. Genomic mutation. Change in chromosome number, polyploid and aneuploidy. A change in the total number of chromosomes such as polyploidy or aneuploidy. Properties of mutation, FPSC important. So pay close attention to these traits for MCQ section. Mutations are random and non-directional. Rate increased by mutagens, UV, X-rays, and chemicals. Most mutations are recessive and harmful. Neutral mutations may become beneficial under changed conditions. Mutations provide raw material for evolution. Beneficial mutations are rare but crucial for adaptations. Genetic drift and gene flow. These are often referred to as random evolutionary mechanisms because unlike natural selection, they don't necessarily favor the fittest traits. Let's break down these two forces. First, we have genetic drift. Random change in allele frequency not due to natural selection, significant in small populations. This is a random change in allele frequency that is not driven by natural selection. It is most significant in small populations where a single random event can wipe out a specific trait forever. There are two classic examples we must know for the test. Founder effect. A small group leaves main population to start new colony, limited gene pool, high frequency of certain alleles, example high rate of rare diseases in isolated communities. This happens when a small group leaves a main population to start a new colony because their gene pool is limited. Certain rare alleles can become very common. A famous example is high rate of rare diseases in isolated communities like the Amish. Then bottleneck effect. Sudden reduction in population size due to catastrophic event. Survivor genes represent only a fraction of original diversity. Example, cheetahs genetically very similar to past bottleneck. This is a sudden drastic reduction in population size due to a catastrophe like a natural disaster. The survival only represent a fraction of the original genetic diversity. So on the other side we have gene flow or simply migration. This is the transfer of alleles from one population to another. So the There are two main points which we are going to discuss to understand the gene flow effect on diversity. Increases genetic variation in receiving population, reduce variation between populations. It increases genetic variation in the receiving population but actually reduce the overall variation between different populations, making them more similar. Effect on evolution counteracts genetic drift, it prevents interbreeding depression. Think of it counteracts genetic drift and help prevent interbreeding depression. So here are two examples. Example pollen. Pollen carried by wind or insect transfer allele between plants. Population migration. Animals migrating introduce new alleles. For example, bird migration pattern. So think of pollen being carried by wind to a distant forest or bird migration patterns introducing new alleles into a local flock. Understanding the difference between the randomness of drift and the moment of flow is key to mastering this section of the syllabus. Evidence of evolution. Theory is one thing but evidence is another. Scientists use multiple lines of data to confirm evolutionary relationships. Let's break down the 6 pillars we need to know. Fossil record. Preserve remains of past organisms. Show gradual changes over time. Transitional fossils. Archaeopteryst, reptile bird, Tiktaalik fish, land vertebrates, carbon-14 and other radiometric dating. Biochemical evidence, DNA sequences compare between species. Humans share 98.8% DNA with chimpanzees. Cytochrome C protein almost identical across mammals. Universal genetic code, same codons for same amino acids in all life. Comparative anatomy, homologous organs, same structure, different function. 4 limbs of humans, whales, bats, horses. Analogous organ, different structure, same function. Wings of birds, us insects. Convergent evolution. Vestigial organs, reduced non-functional organs, human appendix, whale, pelvic bones, ear, muscles. Biogeography, species distribution patterns match evolutionary predictions. Island species often resemble nearest mainland species. Darwin finches 14 species on Galapagos, adaptive radiation from common ancestor. Embryology, embryos of different vertebrates look similar in early stage, von Baer law. Okay, gill slits tail notochord appear in human embryos proposed by Ernst Haeckel. Ontogeny recapitulates phylogeny, biogenetic law, no dispute in strict form. Direction observation, antibiotic resistance in bacteria, pesticide resistance in insects, industrial melanism, peppered month Western Betullaria, most direct observable example of natural selection. Now let's discuss about speciation. Speciation is the formation of new species. We have discussed how traits change, but how do those changes result in the entirely new species? This process is called speciation. Speciation is the evolutionary process by which populations evolve to become distinct species. Reproductive isolation is the key prerequisite. So there are 3 types of the speciation. Let's discuss one by one. First is allopatric speciation. This is the most common form. A geographic barrier like an ocean or mountain range physically separates a population. Over time, independent evolution leads to reproductive isolation and eventually new species. Darwin finches are the best example here. Darwin finches separated by the Pacific Ocean between islands are the classic example here. Sympatric speciation. This is more complex because new species arise within the same geographic area without any physical barrier. In plants, this often happens suddenly through polyploidy. A famous animal example is the apple maggot fly, which begin diverging based on which fruit they choose to lay eggs on. Third is parapatric speciation. Here populations are adjust to one another with only limited gene flow because they face different selection pressures across their range. They gradually diverge. We can see this we can see this in certain grass species near mining sites that have adapted to soil with high metal content. Isolating mechanism. Prezygotic habitat, temporal, behavioral, mechanical, gametic. So to keep these species separate, nature uses two types of barriers, that is prezygotic, these prevent mating or fertilization from even happening. Think of different habitats, mating seasons, temporal or incompatible mating rituals, behavioral. Post-zygotic, these occur after fertilization. The most famous example is the mule, a cross between a horse and donkey, which is healthy but suffer from hybrid, meaning the two parent species remain distinct. Now let's discuss about Hardy-Weinberg equilibrium. Population genetics. The Hardy-Weinberg principle states that allele and genotype frequencies in a population remains constant from generation to generation in the absence of evolutionary influences. What is the equation? P2 plus 2pq plus q2 is equal to 1, where p is the frequency of dominant allele A, q is the frequency of recessive allele A, and p plus q is equal to 1 is total allele frequency, that is 100%. P, P would be the homozygous dominant AA, 2pq heterozygous A small a, q2 homozygous recessive small aa. What are the significance and applications? Provide a null model. Deviations from HW indicates evolution is occurring, used to calculate carrier frequencies. For example, cystic fibrosis show that dominant allele do not automatically increase in frequency. 5 conditions for HW equilibrium. HW equilibrium is Hardy-Weinberg equilibrium. No mutation, mutation rate is 0. No natural selection, all genotypes equally fit. Random matching, panacea, no gene flow, no immigration and immigration. Large population size, no genetic drift. So here is a comparative summary of evolutionary theories, a quick revision for FPSC. Here are the theories. Lamarckism, proposed by Jean-Baptiste Lamarck, year 1809. Core idea: use and disuse, inheritance of acquired characteristics. Key weaknesses disproved by Weismann, no genetic basis. Second is Darwinism. Charles Darwin A. R. Wallace proposed it in 1959. The core idea was natural selection, survival of the fittest. Didn't explain mechanism of heredity. Mutation theory, Hugo de Vries, 1901. The core idea: sudden mutation drive evolution. Key weakness: mutations alone insufficient, most harmful. Theory Neo-Darwinism, proposed by Fisher, Haldane, Wright and Mayer from 1930 to 1950. Natural selection, Mendelian genetics and population genetics. The key weakness: does not account for epigenetic horizontal gene transfer. Synthetic theory, proposed by Dobbs, Haskell, Mayer, Simpson from 1940 to 50. Unified synthesis of all evolutionary mechanisms. Key weakness: ongoing refinement, extended evolutionary synthesis. So this is the comparative summary of evolutionary theories. To conclude our deep dive, let's look at the two patterns of evolution that are easily confused: adaptive radiation and convergent evolution. Adaptive radiation, which is also called divergent evolution, this is when a single common ancestor evolves into many diverse species to fill different ecological niches. The classic example is Darwin finches 14 different species on the Galapagos Island originating from one ancestor. You see this same pattern in Hawaiian honeycreepers, Australian marsupials, and killied fishes, also called divergent evolution. Then convergent evolution, this is the opposite. Unrelated species develop similar traits independently because they live in similar environment. Examples are wings of birds and birds and insects, eye of octopus and vertebrates, streamlined body of dolphin, mammals, and shark fishes. Important scientists, a quick FPSC reference, memorize this table for the MCQs section. These are the names that frequently appear in the match the following or two proposed questions. First is Lamarck, First Coherent Theory of Evolution 1909, Darwin, Wallace, Natural Selection 1858 to 59, Mendel, Genetics Law, Foundation of Neo-Darwinism, De Vries Mutation Theory 1901, Weismann, Germplasm Theory Disproof Lamarck, Hardy and Weinberg, Population Genetic Equilibrium 1908. Dobzhansky, Genetics and Origin of Species 1937, Ernst Mayr, Biological Species Concept. With these concepts and names mastered, you are fully equipped for any evolution-related question the FPSC throws your way. In next video, we are going to practice the solved MCQs for the theories of evolution. Don't forget to subscribe the channel for more videos and practice quizzes based on FPSC preparation. Thank you for watching and stay tuned for more videos. Don't forget to subscribe.